2 million Americans with diabetes mellitus type 2. And the current treatments for that is could be a pump, patch, and most commonly injections. But frequent insulin injections could cause side effects such as infections, pain, and even reduce the quality of life. Not only that, but it could also produce hazardous waste. So as of right now, there is no oral insulin medication, and that's what we are trying to develop and study. So for this, we use polymers as a drug system to overcome premature biodegradation if we use an oral insulin medication. And polymers, why use polymers as a drug delivery system? Well, because polymers are non-toxic, biodegradable, and cost-effective. Most importantly, they overcome, like I said, the premature biodegradation at the gastrointestinal tract. There are many different types of polymers out there, such as synthetic polymers, condensing polymers, but what we focus on is using a natural polymer called fenugreek, because it could also have the ability to lower blood sugar levels. And we decided to use pigs for this research because they are an ideal model of anatomy and function similar to that of people.
I will then begin by talking about some of the procedures. First off, with conducting the experiment, we acquired 12 piglets, 6 males and 6 females, and we housed them at one of our poultry barns here at our Tarleton State facilities and allowed them to acclimate, to get used to their environment, and to become comfortable with frequent human interaction. The pigs were kept at optimal temperatures and fed equal amounts of feed consistently. We then trained them in a sling to get them used to their blood glucose levels being taken daily, as this was going to be frequent. And we collected small amounts of bloods by pricking their ears. We took these glucose levels twice daily before feeding. Non-diabetic blood glucose levels for piglets usually range between 70 and 120. Streptozocin is a known drug that kills beta cells, which prevents insulin production and allows us the ability to induce the pigs with diabetes mellitus. I will go further into detail on the following slide. We then prepared the therapy, in which Gus will talk about the preparation and why we use specific concentrations. The therapy was always changing due to the fact that pigs gained weight. 1 milligram per deciliter of insulin was administered per kilogram of the pig's weight. The pigs were separated into 3 different groups and pens, 2 males and 2 females per group. There was a therapy group, which we administered insulin and fenugreek to. There was a polymer group in which fenugreek was the only treatment, and then there was a control. This control group was not induced with diabetes and no treatment was given. This was the baseline for our data. We continued this project for a duration of approximately 2 months, and after the project, we dissected and harvested their organs to check for any obvious systemic damage. I will now talk about how we induce diabetes. So as I said before, the pigs were separated into 3 different groups and pens. After they acclimated to their environment, their baseline measurements were taken, such as their weight in kilograms and their blood glucose in milligrams per deciliter. 8 pigs were induced with diabetes mellitus. They were then anesthetized with ketamine, which was given to us by a local veterinarian. And per literature, we found that 50 milligrams per kilogram of weight is sufficient to kill the pig's beta cells, which inhibit insulin production. Following the procedure, the pigs were closely monitored to make sure there was no complications or issues. We then began to take their blood glucose levels twice daily until we determined that the blood glucose level was 3 times their original baseline levels for 3 consecutive days. For example, if the blood glucose level of a piglet was originally 70, we would need it to be higher than 210 for 3 consecutive days. The reason for this, for allowing the blood glucose to be at a diabetic level for 3 consecutive days, is because after the initial injection of streptozocin, there is going to be a spike in blood glucose, but it will often fluctuate for a couple of days. Once the pig is considered diabetic, we then begin treatment. However, it should be noted that not all treatment began at the same time due to the pigs becoming diabetic at different times. This didn't affect our results drastically because the start of the treatment for each pig began within days of each other rather than weeks.
So to begin the preparation of the therapy, we first gather fenugreek seeds and then extracted them into a mucilage and converted it into a fine powder form. Insulin powder was then grafted to fenugreek powder by microwave irradiation. Microwave irradiation created linkage of insulin to the polymer by free radicals. After obtaining the therapy, precipitate and let it dry in an oven at a temperature of 60 degrees Celsius. We then confirm the therapy linkage with 4-ion transform infrared spectroscopy from the microwave method. And then the needed amount of precipitate was based on the current average weight, as Tracy stated, of all pigs, and inserted it into individual pills as needed. So the fenugreek plus insulin treatment, which was known as the therapy group, we used a 1:1 ratio of one standard unit of insulin to 1 kilogram of weight of the pig. And from there on, we used a 4:1 ratio of fenugreek to insulin. Following the fenugreek only treatment group, fenugreek was equal to the amount used in the therapy group. So if for the therapy group we were to use 7 grams of insulin plus polymer, we used 7 grams of fenugreek only for the treatment group, and that's how we did it for 30 consecutive days. So as per the results, as I stated, we did the therapy for the pigs once they had those levels of diabetes. And we've, like, as you can see over here in this graph, the baseline was between 70 to 100 of the insulin levels. But after 30 days, we noticed that mL's blood glucose decreased from diabetic to normal levels. So this is what we wanted to see. And you can see that on the on pig 79 male on the yellow line right here. So it started at a baseline, and once it got diabetic to over 400, and then after the 25 day, it decreased back down to that baseline that we wanted. Also, a female's blood glucose decreased from diabetic to pre-diabetic, and a male's blood glucose remained at a diabetic level. So the female that went from diabetic to pre-diabetic was seen on pig 64 on the blue line right here. So it had the same baseline, and then it increased. Actually, it had a big spike to over 600, a level of 600, and over here it went back down to pre-diabetic. And then it was interesting to see the mL's blood remain at the diabetic level. Following the polymer group only, mL's blood glucose levels returned to normal levels, and mL's also mL's blood glucose levels remain at a diabetic state. So also both females returned to pre-diabetic levels. So this was very interesting to see that that from the polymer only group, and that could be from many reasons why they stayed at those levels. It could be due to human error, or it's just the polymer as that it was. For the control group, all the pigs' blood glucose remained constant. And you can see that on this graph right here. So there was no, like Tracy said, there was no inducing of the pigs there, or no treatment were given.
Going off of the conclusion, after we analyzed and dissected the organs, we concluded that the polymer had no toxic effects due to the lack of systemic damage, thus showing promising results for the continuity of this research, along with any future non-toxic insulin medication delivery. As the results showed there were some differences in the data between the sexes, and we'd like to, in future experiments, we'd like to see if there's a correlation between blood glucose and hormones such as estrogen and testosterone, allowing us to continue the investigation for why the reasons of the results in the males were, the results of the females and males were different. We'd also like to increase insulin concentration in the therapy to show if there are if changing the dosage will give us better and more consistent results. And lastly, we'd like to analyze the tissues and the cells to see if there's any negative side effects on a histological level.
And we'd just like to acknowledge our faculty member, Dr. Rajani Srinivasan, and the College of Agriculture and Natural Resources for their mentorship at Tarleton State University. Thank you.
Thank you guys so much for that. That was a very insightful presentation. Judges, do you guys have any questions for them?
I have a few. First of all, is the point of this to have insulin constantly present in somebody's system? Or would you be taking this drug, or would you be taking this pill after eating or something like that?
So what our main aspect for this research was to like potentially use a pill when needed, or per se like every 24 hours to reduce your blood glucose levels back to normal range. So you'll be still, like, let's say if, for example, if a person was needed to reduce his blood glucose levels, it could, like, they could only like take a pill a day, for example.
Okay, so is that, I know with your pigs, you probably had a very controlled diet. You knew exactly how much carbohydrates they were having and everything like that. For if you were to apply it to humans who eat a variable diet, you eat different amounts of food every day, your blood glucose should fluctuate at a greater rate depending on your activity in your day-to-day. Would you still be able to take only one pill, or would you have to do some type of math to make sure you're getting the same amount of insulin that you need?
No, you wouldn't. I would say you wouldn't take one pill. For this research, since we're using pigs, maybe, but like when it comes to like humans, like you might need to take like 2 pills or like 3 pills depending on the on what you eat and your healthy lifestyle and such things like that.
Okay, that is what I had. It was good research, thanks.
Judges, do you guys need more time to make any final notes for grading or anything like that?
I think I'm okay.
Okay, cool. Audience, do you guys have any questions for them? Okay, great. We can move on to the next presentation. I'm gonna ask Tracy, can you unshare your screen? Thank you so much. The next presentation is Ayush from UT Austin. Whenever you're ready, go ahead and share your screen and you can start.
Well, can you all hear me by chance? Cool, thank you so much. Okay, hi, my name is Ayush. I am an undergraduate research assistant at the University of Texas at Austin. I work at Dr. Fast's lab at the College of Pharmacy. And for the past like 5 to 6 months, our lab has identified and characterized 2 separate inhibitors, one of which is called methylpicolinimidate, and the second one is the one I personally characterized and identified, and that's called 1-chlorisoquinoline. And these 2 inhibitors are important because they inhibit an enzyme called New Delhi metallo-beta-lactamase. It's an enzyme that confers antibiotic resistance to bacteria. So before I talk about the inhibitor at all, I just wanted to give like a background about why antibiotic-resistant bacteria are so dangerous. So according to the Antimicrobial Resistance Review in 2016, approximately 700,000 people died to antibiotic-resistant bacteria. And this is a conservative estimate because it's really difficult to tell. And this number is only expected to increase. In 2050, it's meant to surpass the deaths caused by cancer. And that's like pretty scary. So I want to switch gears and talk about, okay, what are the antibiotics that we have right now? And we see that 60% of the total antibiotic sales worldwide are beta-lactam antibiotics. They're a specific class of antibiotics. And they're called that because they have a characteristic lactam ring. It's the cyclic amide group. And that would sort of beg the question, if there were an enzyme that could break or cleave this beta-lactam group, we'd be in big trouble because that's comprises 60% of our total antibiotics that we have right now. Well, it sort of turns out that these enzymes do exist. They're called beta-lactamases, and there are two sort of broad classes that our research group looks at. The first one is called serine beta-lactamases, and they're the canonical sort of mechanism that we see here. You see that serine residue acting as a nucleophile and attacking that amide, or that cyclic amide, that lactam ring. But you see a few differences with metallo-beta-lactamases. And metallo-beta-lactamases are what we're interested in because they have these zinc cofactors which essentially stabilize that nucleophile and allow it to attack. And it's not there in serine beta-lactamases. And the other sort of big difference is the fact that no covalent adduct is formed. So it's really difficult to form an inhibitor, or at least slightly more difficult to form an inhibitor without that covalent adduct, without something to sort of target, because the interactions are so fleeting. And NDM, a specific type of metallo-beta-lactamase, is what we're interested in because of how widespread it is, in addition to the increasing proportion of carbapenem-resistant bacteria that have NDM. I mean, it's like the trend here, you can see right over there, it's pretty astounding. And carbapenem-resistant bacteria especially are pretty scary because carbapenems are considered our last resort antibiotics. And if NDM breaks those, we need a drug for it real quick because there's no FDA-approved solution or drug for this. So this is where NDM comes in. There are two ways to bind to it, our lab hypothesizes. There are two zinc cofactors where you can non-covalently bind it, and there's a lysine, this K211, I don't know if you can see my mouse. This lysine gets deprotonated and acts as a nucleophile. So if you have any sort of electrophilic moiety in your inhibitor, you can form a covalent bond. So this is where I came in. I did a lot of high throughput screening. These are inhibitors or sort of fragments of inhibitors based on an inhibitor, MEP, methylpicolinimidate, that we found earlier. And these can either covalently or non-covalently bond. We had really no idea. We just wanted to see. And so I did something called a time-dependent inactivation. And pretty much what this is I wanted to see how the inhibitor interacted with the enzyme over time. And I did this at excess zinc concentration because that would decrease the probability of having something called a metal chelator. And chelators are not really that helpful because, as you can see, the enzymes in our body have a lot of metals. And if this were to non-selectively chelate all those metals, we'd have a big problem. And lastly, I wanted something at high inhibitor concentration because I wanted to see, was there any sort of inhibition at a really high concentration? And then I could lower it as I went along, and I found something. And it turns out I did find something called hit number 13, 1-chloroisoquinoline. And this is a time-dependent inactivation assay. And I see that there's a stark difference between the control and hit number 13. But I still had a lot of questions. You know, is this a non-covalent mechanism? Is this covalent? I know there's inhibition because this is really drastic. So what I thought I should do is lower the concentration of the inhibitor from 5 millimolar to 150 micromolar. And I did this because I wanted to see, was that high concentration masking anything? Was the enzyme just being saturated with a lot of inhibitor? I just wanted to see what was going on. And I see here that there is an equilibrium around 70% enzyme activity for the 150 micromolar of 1-chloroisoquinoline. And that tells me that there's a non-covalent reversible adduct formed with NDM. because of this equilibrium. If this were to be covalent, there'd be a permanent decrease in activity, and that's not what we see here. Now, in terms of potency, I wanted to see how strong this thing was. And I did something called an IC50 assay, a lot of assays, but you know, you get through it. And essentially all this tells you is that at 8.6 micromolar, 50% of the enzyme activity is reduced. So if you think about it like intuitively, a lower IC50 means a more potent drug. And to put into context, this is very potent because you have publications that are around 100 micromolar, and this is a lot smaller and thus more potent. But there's still a lot of optimization to be done. Chlorine in isochloroquinoline, it's not the best leaving group because it's not very selective, and it also leads to solubility issues, if you know, like stuff like silver chloride and stuff like that. And our body is made of water, and if this is insoluble, it's really a no-go. So I wanted to replace some of these, the chlorine with carboxylic acid and hydroxyl group. And I did sorry, I flipped that on accident, my bad. But yeah, so I did a docking simulation of just like where this analog would bind, and as you can see here, it binds perfectly right between those zinc cofactors, which is what we were thinking of before. So this sort of confirms that. And lastly, this is sort of a really big picture thing of what we want to do. We want to really optimize inhibitor with both a covalent modifier plus the metal binding group that I was modifying before. And this covalent modifier I mentioned a little bit in the beginning is I wanted something electrophilic enough for this lysine to attack it. And so having both a covalent and non-covalent moiety allows greater specificity in that NDM active site, and it also leads to greater potency, which is what we're looking for all the time. So those are sort of our next steps. And so, yeah, I just wanted to thank everyone who helped me with this project. Dan, my graduate student, who was really helped me out just doing all the assays, or he taught me how to do the assays. And Dr. Alex Ahn, he synthesized all the inhibitors, and Dr. Fast for just his guidance because he's my PI. And lastly, all of you for listening to my presentation. Thank you.
Thank you so much for that. That was a wonderful presentation. I definitely learned a lot. Do the judges have any questions?
I have a quick question. So you were drawing up, I think one of your last slides showed coordination. You think zinc is coordinating to the oxygen atom of that hydroxyl?
Yeah.
So do you not think the nitrogen is participating at all?
It's actually a bit of both.
So
that nitrogen lone pair, yeah, it coordinates right between those two. Yeah, I should have been more specific about that.
It's like between the two zincs, you think?
Yeah.
Okay, that makes sense.
It's really cool.
Yeah. Thanks.
Yeah.
Audience, do you guys have any questions? Okay, cool. Before we move on to the next presentation, I saw that one of our judges just joined. Can I ask you to introduce yourself, please?
Hello, my name is Lassane. I am a grad student in the pharma group, and I'm really sorry that I came in a little late. There was a bit of a situation at home, but yeah, excited to be part of this. Thank you.
That's great. You have your Google form ready, right? The one used to judging? Yeah. Okay, perfect. Okay, we can move to the next presentation. Joe, Blake, I know I talked to you too. Is Nicholas here?
He's not here. He's at another presentation.
Okay, is he going to be here? Should we wait on him, or are you guys gonna present?
No, he's not going to be here for a long time.
Okay, that's fine. So you guys are up next. This is Joe and Blake from Tarleton State. Welcome. You guys can share your screen and start presenting whenever you're ready.
Thank you. Can you guys see it? Okay, I can just make it here. Okay, I'm Joe Becerra. I'm an undergraduate biochemistry major.
I'm Blake Sarman. I'm an undergraduate professional chemistry major.
And we will be talking about the innovative and cost-effective methods of detection using plant-derived polymers for the treatment of microplastics in groundwater.
The research goal for our research is to commercially treat water using organic polymers to remove microplastics and other contaminants.
What are microplastics and why are they dangerous? Microplastics are just plastics that are no larger than 5 millimeters, meaning they can only really be seen through a microscope. And the reason why they're dangerous is because they're small enough to bypass the human natural barriers within the human bodies and other organisms. This means they can eventually accumulate in the body and prohibit or incorporate themselves into cells, causing cell death. Another issue with them is that they can be carriers for other toxic materials. Where do they come from? Microplastics originate with us to two means: to personal use, like plastic bottles, plastic bags, and throwing these away and not recycling them. Another way is through industrial activities, such as the 27% of the microplastics in the ocean come from the textile industry. So as these plastics build up in landfills, they are eventually broken down by the UV sun mechanisms and other microorganisms. As these plastics are degraded, they are washed off into the oceans through runoffs. These microplastics are then ingested by fish these microplastics are also as they are incorporated into phytoplankton, which are eaten by fish. The fish acts as the nexus to where all the plastics go into other organisms. As you can see, aquatic life, seabirds, and even us. And as this happens, we are creating more microplastics. For our current research, we used 4 food-based polymers: fenugreek, okra, tamarind, and taro. These are just the molecular structure of these respective polymers.
The advantages of using our polymers are that they're eco-friendly, water-soluble, biodegradable, non-toxic, and cost-effective. To make the polymers we use the plants. We get the plants and we extract the polymers using like squeezing or grinding, and then we dry them into a fine powder to put in the water. And then as the polymer goes in the water, we remove the microplastics through flocculation, and then we use a microscope and a heatosianometer to count the microplastics for our experiment.
For the water we use for our research, Southwest Lubbock area, well 15, 16, 17, and we got those from the High Plains Underground Water Conservation District.
The jar test we use to flocculate our samples. So as you can see, there's 3 jars. One is for the control, the polysaccharide, and then a comparative sample of polyacrylamide, which is used in the industry currently. We run the jar test for 1 minute and then 5 minutes, and then we let it settle, and then we take the samples at different times. And using the heatosianometer, we usually use the 4 corners, and we find the pieces of microplastic. We count them, and then we use this equation seen right here to calculate the percent removed compared between the control and our polysaccharide sample.
And so we also, while we're counting these, we also look for, we also go to document actual proof that these polymers are grabbing these microplastics. So as the image you see, slightly blurry, is the tamarind polymer that has entrapped 4 microplastics into it. 3 are obvious, the top 3, and then the bottom one is kind of hard to see, which does lead to some issues that you can see in the results. In the results, they typically follow this similar pattern. The controls for each time interval has the highest amount of plastics.
The polymer or the polysaccharide and the polyacrylamide typically are comparable to each other, which you can see in the bottom table. Polymer starts at 35, goes to 46. The polyacrylamide starts at 27 and ends at 53. And the chart on the right is just to visually represent the first table. This was done with the fenugreek polymer at the dosage of 1 gram per liter at well 16. This next table was for taro polymer at a dosage of 1 gram per liter at well 15. As you see, similar pattern. Control has the highest amount of plastics, the polypolymer and the polyacrylamide relatively low. However, you can see there is a spike at the 60-minute mark, and that can be due to two factors: human error and the concentration of microplastics within a certain area. So when we take the sample for the hemocytometer, we only take 10 microliters of a 20 milliliter sample that we collect. So there's going to be spots where there's high density of plastics and low density of plastics. We've tried to standardize this, but it's been in some trials. And that's why you see a negative 44% for the second table at the bottom. This is the tamarin polymer, which is done at the same dosage, however, in the same water as well. You can see that the top table is relatively the same pattern, except for when you notice at the second table, it's negative percentages for both of them. That is due to the same factor of low density and high density in certain areas. As you can see, the control has low density of polymers for the 60-minute mark, even though all of them are going down. As you can see, the initial marks for polysaccharide were around 1715, polyacrylamide was 1813, and they ended up 5.3 and 7.4. And this is this is a combination polymer, which is at 1 gram per liter. So 0.25 grams of okra and 0.25 grams of fenugreek into 100 milliliters, which goes into the jar test. Same dosage, same water. You can see the same issue what happened with the tamarin earlier, or the taro happened earlier. Either human error or they just picked up a large group of plastic concentration.
We also use ion chromatography to see if ions such as anions and cations are removed during this process. As you can see, so taro was able to remove a lot of the fluoride compared to the polyacrylamide. So some things are better at being removed with polyacrylamide, some things are better to be removed with our polymers, and some of our polymers are better at removing things than other of our polymers.
So in conclusion, we have determined that the most optimal dosage is around 1 gram per liter with our current procedure. We also, in past research and current research, we have determined that polymers are also versatile. They can be used to clear out multiple contaminants at the same time, like microplastics, crude oil, suspended solids, and dissolved solids. And another benefit is that these microplastics, these polymers can easily be incorporated into existing water treatment infrastructure, as it's the same procedure to use these as polyacrylamide, and they're comparable with polyacrylamide. I would like to acknowledge the High Plains Underground Water Conservation District for giving us the water that we use, the Robert A. Welch Foundation for the departmental grant, the National Science Foundation, REU, the Endowment Bernardine Munson Foundation, the President of the Isolation Research Scholarship, and special thanks to our colleague Nicholas Ramos, and our faculty member and mentor, Dr. Srinivasan. These are the references for the images I use. Any questions?
Yeah, judges or audience, feel free to ask any questions at this time.
I had a question. Was wondering what your future plans are after capturing the microplastics. What does your research group think is the best way of getting rid of them finally?
We are currently just focusing on figuring out which combination of polymers would be best at removing the most amount of plastics.
What we do afterwards would be for we would have to look into.
Do you have any future plans to correct the discrepancies you're seeing in your final time point to kind of figure out how to correct the human error or that distribution of microplastics?
Yeah, we currently we're just what we're doing is before we get to 10 microliters, we're stirring it to try to make it evenly distributed. We had also had originally had tried a dyeing procedure to dye microplastics, so there's no like you can't question which is what is what. However, the issue we ran into that was that certain of the polymers we were using would be dyed by that certain dye. So we're looking into that.
Dye we used was mallow red, by the way.
And following up on that, it sounded like you were taking aliquots at different time points. Are you taking multiple aliquots at each of those time points?
Yes.
We take duplicates at each time point. So we take 4 time points, and then each time point has 2 duplicates. So there's 8 total. In each the control, polyacrylamide, and polysaccharide, which is our polymer.
Thank you.
Welcome.
Okay, I'm assuming there's no more questions from anyone. Judges, do you guys need more time to write feedback or comments on this presentation? Okay, that's great.